Keep it simple. There we go. Hi, Chris.
Good morning.
Good morning.
How's the temperature in Bordeaux?
It's cold.
It's
cold. It was snowing a couple of days ago.
I know, I saw.
Yeah,
First time since 11 years.
Bordeaux. Did you get the day off? Just because it's so rare.
Yeah, everybody was excited.
Good. As you should be, especially on a Monday morning at 9:30. So welcome to Neuroccino. Thanks for sharing the link, Victor. So as you can already see, Victor is presenting a paper today on resting state. That's all he told me about it for now. The way it works is everyone is welcome to join us and present a paper or a piece of science they got excited about, and just come and pitch it to us and tell us why you were excited about it. So without further ado, Victor, if you want to share your screen.
All right.
Exciting resting state paper.
Okay. Can you see it well? No. There is the bar from the. All right.
We can see it just fine.
All right. Perfect. So the article I'm presenting today, as you can see, entitled Robust Dynamic Brain Coactivation States Estimated in Individuals. Well, it got me pretty excited at first when I read it, when I read the title at least, and where I saw it published, which it is published in Science Advances and will be in the issue for next month, I think. And it's about, like, as a lot of articles, and it's a whole field of research, right? The resting states analysis, the resting state networks, understanding how resting state fluctuation, interesting fluctuation in the fMRI signal means something, how it organizes the brain. And it's been about a decade, I'd say, since when we started to explore the dynamics of resting states through basically, instead of looking at resting state networks on the whole fMRI acquisition, saying, okay, what correlates with what in the brain, it was done by using like sliding windows to see what correlates with what at a specific time point in the or time window during the acquisition, and see how it evolves along the scan to see really the dynamics of things happening here. Well, this article says, yeah, but it's not good enough because you don't have, like, you're still limited to a time window, and so you limit your resolution in time. So what we want to do is to have something that can characterize the state your brain is in at the single time point resolution. So, and to characterize it at the individual level, because this is not another problem. A lot of time, because the signal is really noisy and it's fMRI, right? So it's quite hard to get a proper organization of the brain, to estimate the proper organization of the brain at the individual level. You can get some really, really nice results at the group level, but when it comes to individuals, it's a bit harder. Well, sometimes it's very hard, and so it limits our ability to characterize the brain in terms of everything that is related, for example, to the clinics, which is really too bad, because it's a like resting state is really nice for the clinics, at least in theory, because you don't need to put someone to have someone to do any kind of task. You just put them in the machine, and that's it, basically. Sorry. So they design a method here it is, to estimate what they call brain states for each single time point in an fMRI scan. So how they do it is they start with the brain scans of a lot of participants, and they use a parcellation atlas, the Yeo atlas, that is very well known in the field, but is for me it's a bit coarse. But well, anyway, they use it to estimate for each time point. They associate one patch of the atlas with the mean value in this time point of this subject. So you and then basically they binarize it between either -1 or 1 if the mean result for like this patch is over or under 0. So you, in the end, you end up with some kind of black and white or like -1 and 1 map per time point, per subject, telling you if the patch is activated or deactivated, basically. Then they took all those maps and then just did a big k-mean clustering into 16 different clusters to obtain the brain states, which give you all the different clusters that show like specific pattern of activation plus deactivation in the brain. So like, for example, here they show like 1, 2 to 16 and saying, okay, so we have one cluster of things that activate here, there, and there, which like this one looks really like the default mode network, and then they have all the others too. So that you have a set of brain states defined at the group level, and then you want to estimate this in terms of individual levels. How do you obtain this brain state at the individual level? Well, you basically repeat the same thing. You take the atlas for each patch of the atlas, for each region. You associate the mean the mean of the values inside of the patch, and then you obtain a new map for each time point, which is related to this atlas, and you just for each time point you just check which one to which brain state it is closest to, and say you say, okay, this one is closest to brain state 1, so it's brain state 1, then this one and this one. And the main thing that they look into during the article here is the frequency of a brain state during a scan of one participant. Says, like, for example, here you'll have 12%, like 12% of the time you'll have brain state 1, 10% of the time you'll have brain state 2, etc., etc. And so this gives you a histogram that you can then compare between subjects and try to see if it's repeatable and everything. So the method itself here, it's I like it in the fact that it is pretty simple and really easy to apply to new data. In general, the article, I like the part of the article that is about using this data on a lot of different a lot of participants and a lot of different data sets. they do a lot of they do test retest. They really try to go all the way through to characterize their method and to say why it's good. They also do some clinical data analysis. So just to show you what they do here, they show here that the intra-subject variability is basically lower than the inter-subject variability. They also show that depending on the number of time points that you take, you can increase the similarity between the frequency of brain states. That's actually this is probably it's probably the figure that I find the most interesting in the whole paper. To me, it's saying if you want to have a reliable estimation of the states in your brain, you need to do at least probably like 20 minutes of data recording. If you look at the inflection point or something like that here, or 15 minutes, because this is where you start to really get a better like reproducibility of your data, even inside of one subject or at the inter-subject level too. So there is really a big gain going from 5 minutes to 15 minutes, which is interesting, but also which means that it kind of sucks when you want to do a quick patient in a machine and you want them to be as little stress as possible. You can't just do it just 1 5-minute resting state acquisition, because you'll get some data, but that won't be super great. But at the same time, you can see like after 25 minutes, you kind of reach a plateau. It keeps increasing, but not that much. So there is really a sweet spot around 15 to 20 minutes. So that's the thing that I found the most interesting in their analysis personally. And so they also selected the they want to prove the effectiveness of their method. They focused on the analysis of interhemispheric differences and to check this the lateralization of the brain, and to do that they selected the 2 brain states where they showed the highest lateralization, with, like, for example, brain state 15, which showed much strong activation in the left hemisphere and rather strong deactivation in the right hemisphere. And so they showed that it is related to task fMRI during a language task. And well, it kind of makes sense because it really looks like a language network what they have here. So they also do, this is again, so they also do characterize these 2 networks like 15 and 11 in terms of other biological measures like handedness and gender and so, or sex, and so they say, okay, yeah, we can find some differences between the left-handed and right-handed people and the male and female with these 2 things, with these 2 brain states. And they also try to do some applications with post-stroke recovery on patients, and saying, okay, if you look at the brain states at just after the stroke or 180 days after the stroke, you can see some changes that are significant and some that are related to recovery, and that's measurable. And so yeah, that's basically the article. They say, yeah, we have a new method. Here is our characterization of the method, and it works. So my main critics of the method is like, I was quite surprised actually that this method is what we have like as the top of the science nowadays. It's not a bad method at all, but it lacks a lot of, it doesn't give as much insight as I would like to into the brain working as what we would expect like nowadays. For example, I have almost there's no explanation about the relationship between different brain areas. The brain areas are quite quite large. The atlas is quite coarse, and they say that they do analysis at the it's estimated in individuals. That's great, but it's because it's based on an atlas at the very beginning. You are removing a lot of variability, and you are actually erasing some of the thing that would be interesting at the individual levels, because you are considering that everyone has the same organization at the start, and that you are starting with that. Plus, it ignores everything about connectivity between regions, either functionally. Well, functionally you can still re-extract it, but it's mostly like any structural connectivity gone. So that's, yeah, that's why I'm like, I have my reserves about this method. I think it's nice. I'm waiting to see what it can give us in the future. Oh yeah, one thing that I really was disappointing that they didn't look at is the chain of different brain states through time. It's like nowhere in the article. Even though it's like the temporal changes between brain states, it's how your all the brain is organized in time. No, I don't think they looked at it at all. So that's a bit disappointing too. All right.
Thank you, Victor. This is great. This is the first time that 3 hands go up even before you finish presenting the paper. Well, let's go in the order I have here on my screen. So Hervé.
Thanks, Victor, for the presentation. So based on what you said at the end, I was also surprised I didn't look at the changes of networks or connectivity and between the different states. And I was wondering, so they did not look at that, and did they look at the time? Because for me, the occurrence rate is not clear for me. Maybe you have more, you can clarify that. Did they look at the time of each brain states? So
No.
maybe the DMN is more.
Yeah, no, I don't think so. Well, I didn't read every single sentence in the article, but as much as I read, it's really focused just on the rate at the global scan level, and not at the when they happen compared to each other. At the end of the article in the discussion, they say, oh, we only look at that. We could have looked at other things, like, for example, the rate of changes. But they say that the rate of changes is really linked to the rates of the thing that happens. So it's not like super interesting. I'm like, yeah, but what I want to know is what goes from what to what at what point, and yeah, the real dynamics, and not just like that. But yeah, that's it.
Maybe they wanted to simplify at the maximum to have one number. I don't know. And I just have a quick follow-up question that is on the. I saw during the method they do a binarization of the maps, and you were, based on what you say, you were pretty skeptical about that. But I was wondering if they do that only to have a clear templates of regions, or there is another reason for doing that?
I don't exactly know why they do it, because I think it helps with the k-mean clustering afterward. and helps having defined brain state instead of a big blur of different clusters that is hard to delineate. I guess that's the reason.
Okay, yeah, makes sense in a way.
Mm.
Okay, thank you, Victor.
You're welcome.
Chris, you also have a question.
Hey, thanks for the presentation. One thing that I'm wondering is how do they explain how do they explain that if it's related to something that exists in the brain, because as you said, they use a lot of ways to summarize the information in a way, because it's atlas based and then they take the mean, so that's a lot of reduction of the actual information you have in the image. How do they know that? So yeah, then you can say, oh, we see a variation in these kind of patients and all, but how do you know, how do they know that it corresponds to something that is actually happening, and if you can predict anything? Because that would be the end goal with this kind of method, no?
Yeah, well, it's just, to me at least, it's just a start for now. They did checked like on the two network, like the 11 and 15, the relationship with task activation of a language task. So they could say, okay, for this one at least, we know that it is related to the language, and that at the individual level it seems to be working, and so yeah, that's
but just for one state, because the other one, I mean, the correlation 0.3 is kind of significant, but it's a correlation to something that is already pretty noisy. So I mean, out of all the other states, I mean, if you take the best state and compare it to all the literature and just find one thing that is similar, that's a little bit thin, no?
Well, yeah, but at the same time, it was mostly one validation as more like a proof of concept, I guess. You'll need to know exactly which brain state does what and relates to what. You'll need to do much more for each single brain state, I guess. Which is actually something of the same problem that I have with the atlas that I'm publishing right now. Is like, okay, we can say this and that, it's like clearly related to motor function of the left and right upper limb. But other than that, we have also other things that are like strange and harder to explain. So yeah, for me, they managed to show pretty convincingly that they are encoding some interesting information at the individual level. I don't know if it will work for every individuals, and I don't think so, at least actually. But I think they are managing to simplify and extract some interesting information. Problem is, I think it's really too like, for example, here they really show some like large differences occurring across time for these patients. So that was also that you see there is a lot of evidences that there is some nice signal that they encode in them with their method. But I completely agree that it's oversimplifying the complexity of the signal that we get with fMRI, and we are losing so much with this. I'm a bit skeptical on the end game that they will what they can do with this.
All right, thanks.
I can just jump in and come back to the stroke figure that you just showed.
Went too far here.
Yes. So what did they actually describe using their method? In the in anatomical changes in stroke, but also was this correlated with any behavioral or cognitive measures to actually show that these are meaningful changes in recovery?
No, I don't think so. I don't remember. Maybe they did, and I just went through it without reading it. But at least in the obvious result that is in the figure, no, it's just purely the only variable that you have with stroke is the time after stroke, basically. And then you have their measures, and they can see that depending on the time, you'll have changes in their measures, and showing that it's meaningful.
So strictly speaking, there shouldn't be a chart of recovery. It's just time after stroke, because we don't know if they actually had meaningful recovery or not.
Well, I can check for that. Maybe they have they checked it actually and selected only those that recovered on something like that. I don't know.
Okay, I'll have a look. Anna has a question as well.
Yeah, I had actually initially a different question, but now another question based on Chris' question popped up. So I was wondering, they are going more deeply into brain state 11 and 15, right? Like correlating them with language and I was wondering whether they also do that for all the other brain states, because in one of the first figures you showed where you were able to see the occurrence of brain states 11 and 15 were one of the last ones. So yeah, exactly. So I was wondering whether they do it for all the brain states and all the other ones are just
no, on this one
or
no, on this one they really focus on the brain state that are showing strong lateralization. These are the two ones with the strongest lateralization, because for the others, I guess they didn't exactly know how to do it, or they should, for at least in the article, it was really okay, lateralization is a very important component of brain organization and related to how the brain works, and it's been shown in a lot of studies before. So we will focus on this particular brain specificity, and it's really also linked to individuals. So it's a good way to check if our individual scheme is working here. And so this is how they justify it and say, okay, we'll only take the two brain states that are strongly lateralized, so that we can have a good test for to show what's happening here, which is fair, I think, when it comes to introducing a new method and testing it. But it's true that these brain states are really are quite scarce. But quite scarce doesn't mean either that they never happen. Like they happen. It's, if I go back here, it's about 4% of the time there is one of these two brain states. So it's not huge, but on the whole thing, the whole acquisition, still they still do happen.
Right, but if they would have, for example, decided on some additional information than just the k-means clustering, which, as far as I understood, resulted in those 16 states, right? They didn't back it up with anything else. So if they would have decided on, for example, 10 states, then they would have not occurred at all.
Yeah, so actually they tried, well, to select the 16 states, they did something a bit classical, well, where they're trying to find how many clusters is most stable when you do it. And
Right.
so they had I think 3 local minima, which was 10, 16 and 19 clusters.
Okay.
And so they say, yeah, but we selected 16 because the other, I think it was the one with the lowest minimum in terms of stability, well, highest stability, but the local minima in terms of variability. And also, and they said the 2 other types, it's mostly like either combination or segregated, like division of brain state that we showed here. So that's just a different type of, it's like hierarchically organized and differently segregated regions and brain states. So, but this is to me, it's a problem that we see everywhere when you try to do a functional segmentation of the brain anyway. So, yeah.
Okay, thank you.
Michelle, do you still have a question, or did we take too long for you?
I know, because it was time, I thought I'll ask later. But yeah, I have a question. So it's a great paper on enhanced alert questions that are important in the need for, technical progress and individual personal estimation of resting state network. But it relies on a lot of assumptions, and one in particular bothered me, besides the one that have already been mentioned. Is that true that different brain state will follow the same parcellation of the brain, even at the individual level? Let's not even talk about using a general parcellation for everybody, but if you have a parcellation for an individual based on his data, is there no change in the boundaries between regions according to the states? Do we know that, or can can there be like some smooth transition between boundaries and regions? And
yeah, you can completely have that. But that will be erased with the method. That's and you won't be able to see it.
Yeah.
Well, but to be fair, even if you have these kind of things, and that they won't properly show up with this method, you will still have probably some of the information that you lose still ending up in the states that you have here. It will be deformed, it will be incomplete, but it's real. If there is something in like a strong information to extract from that to be used for the characterization of the participants or something like that, it probably will still somehow show up somewhere in the brain state. It won't be the information that you started with. That's for sure.
I see.
And so to me, that's a big loss, because you won't be able to say these participants are special because. But I think it will modify the signal that you get here in a way that would probably be detectable. But still, this is why I'm a bit skeptical about this very simplification. It's like this method that simplifies a lot of the signals, that, yeah, you lose a lot of things.
I see. Thanks.
Final question from me. You mentioned that it takes somewhere, or ideally you should acquire 15 to 20 minutes of data. Could that estimate of time be improved depending on the scanner?
I don't know.
Because you're right, that's quite a long time, especially in a clinical setting.
Yeah, that's here. I really don't have the answer on
Okay.
this. I doubt it, because with this method, at least the way they characterize it, once again, because you remove a lot of the signal with this thing, I think probably, if to me at least, it's complete speculation, but I think if you manage to characterize each individual while keeping as much as the signal as you have, you may be able to characterize the brain and the states with much slower, like a much shorter thing that we have here. But because you are removing a lot of information, you need a lot of repeat time, and you end up with 15 to 20 minutes of scan time needed. But maybe with a good scanner and another method that keeps the signal, like as much signal as possible, you'll end up with something much shorter. But yeah, it's speculation. It's not there yet. So, yep.
Lovely. Thank you so much for presenting the paper. Thank you all for being here and indulging in this discussion. I wish you an exciting week in science and see you next Monday for the next Neuroccino. Bye.
Bye.